Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to thank Dr. Robi Nur Cahyono, MSc, as the lecturer of the Undergraduate Thesis Seminar course for the time and opportunity you have given me to present my final research result. My name is Farahanisa Zuhdi Iska and would like to present my undergraduate thesis entitled Hirsutrain as a candidate for COVID-19 antivirus based on molecular docking studies, which is supervised by Prof. Dr. Harno Dwi Pranowo, MSI, and Dr. Winarto Haryadi, MSI. The background of this research was derived from the new coronavirus that was discovered by the end of 2019 in December. The new coronavirus is also called a SARS-CoV-2 or Severe Acute Respiratory Syndrome Coronavirus 2, where Shirin et al. 2020 proposed that the new coronavirus is phylogenetically similar to the SARS-CoV-2 that was discovered many years ago, along with Middle Eastern Respiratory Syndrome coronavirus. These coronaviruses infect mammals, birds, chickens, and also humans, from mild respiratory infections in animals and to deadly respiratory infections in humans. Although there are already few drugs that have been approved by FDA, such as Paxlovid, however, there are still several concerns, including the growth of bone and cartilage, and also the consumer is only those above 18 years old. In addition to that, mutation of the virus is still overwhelming currently, as there are many variants introduced since the 2019. Fortunately, research found that chloroquine showed a strong antiviral activity once tested on primate cells, including human, that got infected by the coronavirus. Therefore, there are indications that compounds that have pharmacophore of chloroquine can be a candidate compounds with therapeutic effects of SARS-CoV-2. Hirsutrain is one of the examples, where it has a Latin name of Gossypium hirsutum L. and can be found in Indonesia and has an active compound as an antiviral for COVID-19. Through this compound, then molecular docking will be carried out with several proteins that are responsible for the COVID-19 outbreak. The aims of this research are to prepare ligand molecules of hirsutrain and its derivatives through geometrical optimization. Secondly, to obtain the best docked result from hirsutrain and its derivatives as COVID-19 antivirus through molecular docking with SARS-CoV-2 main protease, and to study and analyze the interaction stability of the best docked result of hirsutrain and its derivatives through molecular dynamic simulation. This is the structure of hirsutrain, where it contains many phenol groups which acts as an antiviral. In 2019, the outbreak began in China, where it caused more than 200 cases and also 3 deaths. And then in 2020, the first case was reported in Indonesia from a 31-year-old woman who had a contact with a Japanese citizen, and until now, at 2022, the virus is still spreading around the world. What are the causes and how does it spread? The causes are acute upper respiratory tract infection and high death risk for risk groups, such as those people who have chronic diseases, diabetes, and hypertension. Research found that the R0 of this virus is above 2, which means that it has an exponential speed of spread. And drugs and vaccines are still under development, although there are already drugs and vaccines who have achieved final clinical trials. Research from Gordon et al. 2020 showed that in total there were 332 types of high-confidence interactions between the 26 SARS-CoV-2 proteins and their human interactors. The red diamond here represents a SARS-CoV-2 viral protein, while the gray circles are interacting human host proteins. The result of this research can be used as a basis for screening active Indonesian natural ingredients for COVID-19. On the other hand, this illustration is a crystal structure of a single chain of SARS-CoV-2 main protease, which indicates the amino acid residues of the catalytic site. And as you can see here, there are green and yellow spheres, where the green one represents histidine 41 and the yellow sphere represents cysteine 145. Why is molecular docking used in this research? It is used to model the interactions of these natural compounds with protein receptor, and the analyzed result are RMSD value, interaction energy, and amount of hydrogen bonds. Molecular docking works by preparing the target protein, and then the target protein and standard native ligands are separated, and then it gets redocked by other ligands. The first hypothesis formulation is from D'Souza et al. 2017, where he reported that DFT calculations for 3 representative polyphenols, epigallocassetin, camphorol, and quercetin were performed using the DFT methodology with the B3LYP functional using the 631GDP basis set, and the conformation adopted by these molecules were assessed. Therefore, if the epigallocassetin, camphorol, and quercetin were successfully optimized at the B3LYP 631GDP level, then hirsutrin and its derivatives can be optimized using the same method. The second hypothesis is that Umar 2021 proposed that the resulting affinity scores among the 3 flavonoid compounds of rutin, four flavonoid compounds of rutin, quercetin, 3-glucoside, remdesivir, and astralgin were the following, respectively. Consequently, the available interactions with amino acids from main protease of SARS-CoV-2 were conventional hydrogen bond, pi alkyl, and pi sigma. Hence, the data suggested that hirsutrin and its derivatives could potentially inhibit the main protease activity of SARS-CoV-2. Therefore, if hirsutrin and its derivatives have conventional hydrogen bond, pi alkyl, and pi sigma interactions with amino acids from the main protease of SARS-CoV-2 after docking, then hirsutrin and its derivatives have potential as COVID-19 antiviral, as they are also classified as flavonoids. The research was held firstly by determining the derivatives of hirsutrin using MolSoft software, where the R here is replaced by H, CH3, COO, CH3, COOH, C6, H11O5, and C7H13O5. And then the molecules were optimized using Gaussian software, using the FTB3LYP method and 631GDP basis set. And then they were converted into .mol2 file using OpenBabel GUI. And then molecular docking was done by downloading the SARS-CoV-2 main protease via PDB with a code of 6W63, where the molecule and standard ligand of the main protease were separated and dock prepped using Chimera. And then the molecule and derivatives were docked using autodock tools, and not forgetting that the grid box was set by 40 times 40 times 40 Armstrong to the power of 3, where the conformations produced gets analyzed its binding energy, RMSD value, number, and also number of H bonds. And finally, the results were visualized using Discovery Studio Visualizer. Lastly, molecular dynamic simulation was done using GROMACS software, where the best docking result was analyzed in it. The force field used was CHARM under condition of 300 Kelvin and 1 atm for 10 nanoseconds, and visualized using the Discovery Studio Visualizer. The result of the geometry optimization is shown here. And as for molecular docking, as you can see here, the standard ligand of X77 consisted many interactions, but according to UMAR 2021, it mainly contains conventional hydrogen bond, pi sigma and pi alkyl, which is found on a hirsuterne derivative that is modified as CH3 with interactions of the same ones as X77. This table here shows that the ligand modified as CH3 was chosen as it has the best docking result overall, although the RMSD value is not the best, but it has the highest binding energy after X77 ligand. This table also shows that the ligand modified CH3 was chosen based on its interactions with X77 ligand that are similar. And lastly, MD simulation. Here there are three graphs produced: RMSD, RMSF, and amino acid residue, and radius of gyration, where RMSD acts as a stability indicator of a protein ligand complex. During 0 to 0.5 nanoseconds, it increased stably until the end of the simulation. From 0 to 0.5 nanoseconds, the graph was low, but then after that, it increased stably until the end of the simulation. As for the RMSF of amino acid residue, the graph was stable at around 0.05 and 0.15 nanometer just after the simulation began and just before the simulation ended. And lastly, for the radius of gyration, the graph shows stability at around 2.24 nanometer, as seen here. In conclusion, geometry optimization was done using the FT-B3LYP method with basis set of 631 GDP on hirsuterne and its derivative, where CH2OH was modified as the following. And then the best docked result chosen was the 7th conformation of hirsuterne derivative with CH3 modification, as it has the most similar interaction types with the standard ligand. Therefore, it has a potential as a candidate for COVID-19 antiviral. And then, lastly, the 7th conformation of hirsuterne derivative with CH3 modification, depicted its stability through RMSD, RMSF, and radius of gyration graphs obtained from molecular dynamics simulation. Thank you for your attention.